All right. I feel I may have rushed you guys a little bit with replication. I just want to make sure you understand that before a cell divides, it all the DNA in that cell has to double itself, has to replicate itself, has to make a copy of itself. So the process of replication is just DNA becoming more DNA, all right? Making a copy of itself. And then a cell divides, and then it does it again, and then those cells divide, and then it does it again, and then those cells divide. How does replication occur? Well, you have double-stranded DNA, you open it up, you unzip it, because remember, it's a double helix, you open it up, and then there's an enzyme that helps to do that, okay, that does that. All right, so you've unzipped it, and then the DNA nucleotides come and they line up complementary base pair to the nucleotides of the strand. So like if there's an A on the strand, a TMP, a DTMP comes and lines up right there. And if there's a G on the strand, then a C is going to come there. A DCMP is going to come. And then once everything's lined up properly, there's an enzyme called DNA polymerase that zips it all up, creates a sugar phosphate backbone, and then your cell divides, and then you've got two double-stranded DNAs in the two new cells. And that's replication. Now we're going to get into the concept of transcription and translation. You've all heard of the term gene. What is a gene anyway? A gene is just a stretch of DNA, and it codes for a specific polypeptide. It codes maybe not for the whole protein, but a portion of a polypeptide. Now, some genes code for a whole protein, some genes code for part of a protein, and then another gene codes for another part of it, and you may need more than one gene to create a protein, or one gene could form parts of many different proteins. So there are about 20,000 genes in the human genome. Back in the olden days, they thought we had about 100,000 genes, but we don't. We have only about 20,000 genes. And they're all interspersed on all the chromosomes. You know that we have 23 pairs of chromosomes. And what is a chromosome? A chromosome is just DNA wrapped with those histone proteins that I told you guys about in the last week. And because DNA is negatively charged because of all those phosphate groups, it attracts the histone proteins that have a lot of histidines in them, and the histidines are positively charged. And so it wraps around and coils, and then it can fit all that DNA into the nucleus of your cell. Okay, so genes code for protein or polypeptides. So how does your body make protein? Well, the instructions are in the gene, okay? All those instructions are in the genes. And what we have to do is get those instructions and make the proteins at the ribosomes. Now, the genes, which are in the DNA, they're all in the nucleus. Remember, if you biology class, here's your cell, here's a nucleus, and this is where all the DNA is. So the DNA is in the nucleus. In the nucleus, this is where replication occurs. Okay, that's where your DNA has to double up before your cell is going to divide. Okay, and in the nucleus, that's where transcription occurs. We're going to get into that in a minute. So replication and transcription all occur in the nucleus. Translation, which is when we're actually making the protein, that occurs in the cytoplasm, at a place called the ribosomes. So pretending this is my cell, in the middle of the cell is the nucleus. That's where all your chromosomes are, your genes are parts of sections of the chromosome. And this is where all the DNA information is, all right? But in order to make protein, we have to do it out here. So how do we get, if the instructions are in the nucleus and yet the proteins have to be made out here, we need a messenger to get the information out of the nucleus and to the ribosomes. And that's where RNA comes in. So the DNA in the nucleus stays in the nucleus. The proteins are made in the cytoplasm, but the RNA is going to come out of the nucleus and over here to the cytoplasm to give the instructions that it got from the DNA. It's really incredible how all this works so beautifully. Okay, so we already know that replication is when you make a copy of DNA. So DNA, you get more DNA. Transcription, the term transcription that I just told you about that is in the nucleus, this is where DNA information gets turned into RNA information. So basically you're making a copy of the DNA, and making that copy is going to be a complementary copy of DNA in the form of RNA. And then that RNA can leave the nucleus and go to the cytoplasm where the ribosomes are, and we can do translation. And what is translation? That's when you translate nucleotide language and turn it into amino acid language. So you're turning your RNA information into a strand of amino acids that will eventually become a protein. Okay, so make sure you know the difference between replication, transcription, translation. Replication occurs in the nucleus, and that's just when DNA makes a copy of itself before a cell divides. Transcription is when your DNA is transcribed, it's copied to form an RNA molecule that leaves the nucleus in order to get that information that it got from the DNA over to the cytoplasm, the ribosomes, where you can make the protein. What is happening there? That process where your RNA is giving information to get those amino acids to make a protein, that's called translation. So you're translating nucleotide language, which is in the RNA, and turning it into amino acid language in order to create your protein. Okay, now, first step is that you're going to have, and we're going to go through the whole process in more detail in the next video. You're going to have a strand, a double-stranded, doesn't have to be wiggly. Let's just take, again, let's say we have this double helix here, okay? We're going to pretend that, sorry guys, I want to make it as realistic as possible. All right, so let's pretend that this section here happens to be a gene. And maybe this section over here is another gene. So a portion, a section of your DNA, that's the gene that codes for some type of amazing protein in your body that makes you super smart, whatever hypothetical, okay? So this is going to be double-stranded, right? So this is, let's pretend that this is my gene, and this is my 5', 3', and this is my 3', 5', okay? One is going to be called the template strand. The other one is called the informational strand, all right? And we're going to get into the details of that. This is going to open up, and then you're going to be making an RNA copy of one of the strands, not both of them. So you're going to go from DNA into something called hnRNA, heteronuclear RNA. And then we're going to cut some things out of that heteronuclear RNA, and then turn it into something called mRNA, which you may have heard of, or messenger RNA. That is, so this part here, when DNA becomes H, or the section of DNA that is the gene, when we make a copy of it and form hnRNA, and then the hnRNA becomes messenger RNA, all that is transcription. Once the messenger RNA leaves the nucleus and goes to the ribosomes, at the ribosomes, there's another RNA there called rRNA, ribosomal RNA, and then another RNA called tRNA, called transfer RNA. They're all going to be involved in translation. Remember how I told you at the beginning of this chapter how there are lots of different types of RNA? So there's hnRNA, mRNA, rRNA, and tRNA. 4 different types of RNA. That would be a good quiz question. How many different types of RNA did we cover? Anyway, so at the ribosomes, that's where your messenger RNA is going to get read by the tRNA, and we're going to deposit the correct amino acids. So we're going to get into all of that. I just wanted to give you kind of a bit of an overview to get you ready for the next step, which is transcription and translation. So I'll see you in the next video.